The broadcast is now starting. All attendees are in listen-only mode.
Hi, everyone. This is Dr. Matthew R. Jorgensen. I'm a toxicologist, chemist, and material scientist with Nielsen Labs, and I'm here with Thor Rollins, our Senior Director of Toxicology and E&L Consulting. We are happy and excited to talk to you about risk assessments for single-use systems. And in this case, through the webinar, we really want to compare and contrast the risk assessment process using USP 665 versus the BPOG protocol. This is something that's become more and more important to us. Manufacturers are using a lot more single-use systems, and us as CROs are seeing a lot more of these come through the lab. People needing help understanding how to set up the testing for this. So I think it's a really timely thing to talk about. But first, what are we really talking about? So we're talking about doing testing on what we call disposables, these single-use systems. These are usually used in the production of a drug substance or drug product, but they can also be used in cases that regulators would consider to be a medical device down the road as well. And this is really popular in biopharmaceuticals. Of course, they're used in other cases, and they're commonly referred to as single-use systems, abbreviated SUS or SAS. And a lot of the motivation for using these is that when you build single-use systems into your manufacturing process, you avoid a lot of heartache and trouble that's associated with validating cleaning processes for contact materials, right? So if you have a bioreactor and you use a one-time use bioreactor bag, then you don't need to worry about how are you going to clean this reactor before the next batch. So really commonly, we have things like storage containers for buffers or intermediates. We have single-use assemblies. This is something that we've seen more and more inside Nielsen Labs, so we've tested quite a few of these. Things like disposable centrifuges. These are all examples of single-use systems that we're seeing come through the lab. So the regulatory framework around this, I think is pretty clear. Of course, we have the US GMPs, so CFR 21165 says that equipment should be constructed so that surfaces that contact components and processed materials or drug products shouldn't be reactive or alter or mess with the API in any way. So basically, we need to know that anything that contacts these drugs isn't going to interfere with the quality or the effectiveness of that substance. In the EU, of course, we have ICH Q7, their version of the GMPs. It really says the same thing. Nothing in that stream, the process stream, should alter the quality of the intermediates or the final API, the final product down the road. So I think that's the charge. We need to be sure that whatever we do doesn't affect the quality of the substance that it's coming into contact with. We know if we are talking about a drug, of course, we don't want something, like a leachable from these plastics to come out and, let's say, react with the API or be there in a way that would block the API from having effect. And that's really what we're trying to prove here. So within the broader framework, so we have the U.S. Pharmacopoeia 665. This is the USP document that covers plastic components and systems used to manufacture pharmaceutical drug products. And then the 1665 is really all about the risk assessment process. How do we decide what parts of a single-use system are high risk and need to be tested? These systems can be large and complex. There can be many steps in the drug manufacturing process, obviously. And so the prospect of testing every piece or every part is kind of a practical impossibility. We really need a mechanism to identify which parts are highest risk and what we should do to address that. So 1665, this is currently in draft. We have an updated second draft that came in October 2020. We're nearing the finish line for this. We hope that this will be officially published really any time now, forward looking to May 2022. So as we go through this, as we start to think about the workflow for evaluation of a pharmaceutical single-use system, we need to begin this process when the design of the single-use system is lockable and ready to prepare for evaluation. So it maybe goes without saying that anytime that you make a change to a device, or you're going to make a change to a manufacturing process, that change is going to have to be evaluated. So you don't really want to start down this path until you know that the process is fixed or lockable enough that you're not going to just have to repeat everything after you make a change. Then we have this single-use system evaluation plan that uses a risk-based approach. That's what we're talking about today, using BPOG or 1665. And then once you've identified the parts that need to be evaluated, and you know the general scheme for how that chemistry testing is going to take place, like extraction solvents, time, and temperature, then you really need to consult a toxicologist. And the reason is that the toxicologist is going to have really specific feedback on some aspects of this, like the sensitivity of the chemical analysis. So it's really important that you know how the toxicologist is going to evaluate these results to make a claim about what compounds are going to need follow-up leachables or not, so that you can be sure that the analysis meets those specifications. Toxicologists can be a really finicky and picky group, difficult to please. And I've seen studies get all the way through this extractables process, and then they go to review by the toxicologist, and the toxicologist is like, sorry, do all of this over, it wasn't sensitive enough, back to square one. And that can be really, really frustrating. It can take a lot of time. So it's good to consult the toxicologist before you really get neck deep into this testing. And of course, after that, the toxicologist will review the data, and if needed, you can go on to a leachable study following that. Most of these actually become resolved as the toxicologist reviews the extractables testing. So at that stage, we often can make a claim about the overall safety of the component and the single-use system, so that leachable studies aren't really required. So I've kind of hinted about this a little bit as I was talking about all the different parts that might be in the manufacturing process for a drug product, where single-use systems can be used. But it's clear this process is long, and the process can be complicated. And as you start to look at each step, the idea of testing every part just like multiplies the burden of this endeavor. So if you take a look at the flow on the screen there, we have this centrifugation, several filtration steps, we have incubation steps, we have chromatography steps, and all of this other stuff. And if we were to do extractables and all of that, it would just be impossible. It's too much to do. So what we really need to do is identify what's the most important thing to test, right? And one of the feelings here is that parts of this process that are most proximate to the final finished form of the device or of the drug product are the most important. So we have this sterile filtration almost at the very end of this flow there, and then we have this final filling apparatus. So we would say, yeah, stuff very far upstream of this final filtration step, if there are extractables, those should be removed through other processes that are part of the manufacturing process. So it's really about like the sterile filtration and the final filling, where we could introduce extractables that might be exposed to the user of the product. And that's, we know that's where we really want to focus. Okay, so this brings us to this idea of, why we want to use a risk-based approach. As we start to set this up, when we make a single-use system evaluation plan, what we'll do is we'll first specify the goal, right? So, we want to evaluate the risk associated with the single-use system for compliance with USP 665, GMP and EU GMP requirements. So it's really important to make sure that that statement is sort of like a scope of work for the document that you're preparing. When you make this evaluation plan, that it's really clearly stated so that regulators understand what the boundaries of this this document are. And then you go through and you perform the actual risk assessment. So instead of testing every single use system in the entire manufacturing flow, we're really only focusing on the ones that have the highest risk of producing a leachable or an extractable that could be exposed to the patient. And then, when you do this, so I said you should know that the design of this process is locked, because any changes that you make could trigger retesting. However, it is good to do this the earliest that you can after locking that flow, right? So it's better to get this figured out before you're really, really far down the path, and it's difficult to adapt if there is ever a situation. So we go through this, we create a list of product contact materials. So we really need to know and understand the manufacturing process from start to finish. Usually this looks like a really long Excel file that has all of the processing steps and all of the contact materials through each step. This should include, it likely includes things like tubing, filters, O-rings are always kind of an interesting piece of this puzzle. And then, this list needs to be all available together with the schematic overview of the production process. And once we're there, right, then we can actually start to quote the cost of this evaluation plan and any testing that might be associated with it down the road. Okay, so from here, I'm going to hand it to Thor Rollins to talk about the general principles of using a risk-based approach in this context.
Thanks, Matt. And as Matt said, we're always excited to kind of talk through risk-based approaches, and when we have to evaluate either drug products or devices that are in contact with drug products or medical devices. It's something we truly enjoy every day, so it's a pleasure to be here with you. With any type of evaluation, so we're going to be looking at both the BPOG methods and the USP 1665 methods to evaluate risks of these sus products. But there's some commonality between those two evaluation types. And so we're going to kind of cover the thought process and when you start doing a risk assessment, either per BPOG or USP 1665, these are things that go into those evaluations. First off, it's material compatibility. Is probably the first big one. And Matt talked about leachables and extractables. How does the material itself hold up to the drug product, the solutions, temperatures, pressures, whatever you're putting it through? How does that impact the materials? Matt talked about this too. This is actually one that may not be as intuitive to some people as it is, but it is about the proximity to the final drug product. When you start looking at the distance along that production stream, that plays an impact on our risk. So the closer it is to the final drug product, the more risky those leachables can be versus something at the very beginning, like Matt talked about. Composition of the contact solution, that's important for us to know when we start looking at testing itself. If there's testing required, that composition is very important. The other thing it's very important when it starts to look at risk is our surface area to volume ratio. So obviously this is a consideration for the amount of leachables that can go into a product. The more surface area to the volume that we have, the more concentrated those leachables can be in that volume. So that's something we have to think about. Temperature and time is pretty straightforward. Obviously, the higher temperatures, the more potential impact. It could be between leachable profiles, and the longer it's in contact with something, the more chemistry comes out, and then any pre-treatment steps. So these are some of the risks factors that we consider. Both approaches, BPOG and USP 1665, tries to evaluate these risks by assigning numerical values to these risk factors and really weighting them based on different, the higher the risk, the more weight they pose. So they really try to help kind of put a numerical value on the potential risks that these systems have to help you understand what you have to do to mitigate those risks. But anytime you're doing a risk assessment, regardless if it's a SUS or a medical device or a drug product, those assessments should be clear and well argumented. They should be thought through. We always talk about a story. You should be able to tell a story of why you're doing a risk assessment, what tools, what factors you used, and what the conclusion is. It really needs to be spelled out. And there are different company-specific approaches, and that's actually okay. That's something that both of these are not necessarily rules that you have to follow. They're useful guidances on how to do a risk assessment. Even though I like the idea of following the USP or BPOG more directly, but that they give you an option of being more company-specific. And then everything's based off the ICH-Q9 management system. So, but the first step in this is if you don't feel comfortable doing this risk assessment yourself, but someone needs to do it. So if you need to reach out to someone to help, we can help, other people can help. But the first assessment really is to make a gap, or the first step is to make a gap assessment to see what information is needed, missing. Like Matt said, you have to walk in this with the information to make it useful. If not, it makes it more painful. So let's actually walk into the BPOG assessment itself. Matt's going to be covering the USP, and we'll compare and contrast the differences. We'll actually use the same example in both, so you can kind of see how the end result may be different depending if you're using BPOG or USP. So in BPOG, like I mentioned, they attempted to assign weighted values to these different factors within the risk assessment. And then, so this is a table out of the BPOG. It's an example. And as you can see, some of the considerations on the left, which is distance along the production stream, like I talked about, exposure, temperature, and the exposure temperature, the duration, the process fluid interaction, and the dilution ratio. All those factors I talked about, yes, on the last page, is things are important. Well, obviously, if they're important, BPOG put it into this table. So these are the consideration factors. And then each one of these factors have ratings. So as you can go down, you look at the different ratings. So let's just take one of them right now, the distance along the production stream. You start looking at different areas. So you start looking at the storage, is it the final formation, the fill, where it's at along those. Or if you look at, temperature, is it frozen? Is it 0 to less than 10 degrees C? So this will help you kind of pinpoint where you think each one of these different risk factors are, and give it a score. And then each one of those scores are weighted based on how we feel or how BPOG feels those risk factors are as far as importance goes. So it's really a mathematical formula at this point. Once you go along and kind of choose the ones you think represents the risk of these subsystems or this component, you go through this risk-based approach, get the weight, and that gives you a mathematical score for that component. So let's take a real-life example here. From Matt's previous example, we'll take the sterilization filter at the end. So as you're going through BPOG, you start seeing, okay, as far as the distance it goes, that's at the final state. So we're going to weight that a 9, or score to 9, and then it's weighted 0.4. So there's your math there. Next is going to be exposure temperature. This temperature for the sterilization is 10 to 30 degrees C. So we chose a 5, and then it's weighted by 0.15. Its exposure time is transient. It doesn't spend a lot of time, less than 60 minutes in there, so it's a 1. As far as the interaction goes, it's a medium. In this particular case, there was a medium solvent when we did this evaluation, and there's a polymeric component, so we used the factor 5. And at the very end, the dilution ratio, it's very concentrated, and so we used the weighted scored 9 with the weight of 0.15. So all those together, if you add up and do the math, it gives you a score of 6.6. So now you can go back to BPOG, and then you have a rating down at the bottom as far as risk category goes. And so we can compare that 6.6 to where we're at on our scoring table. And based on that, obviously, we're in the high risk category, and that means the filter should be tested. We should evaluate that filter for the impact to the drug and the safety of it. So based on the BPOG chart, we're able to identify the risks, and then that tells us basically our next steps. And so you go through this with each component within your system. If it's a low risk, obviously that helps you not have to test it. Medium, based on maybe there's some testing doing. High, we have to test it. So you go through each one that helps you evaluate the risk of each of these components. So that's how BPOG's thought of. I'm going to turn the time back over to Dr. Jorgensen, and he's going to go through an evaluation using the new USP 1665.
Yeah, great. Yeah, and I think that's an awesome overview. The idea with 1665 is really pretty similar to BPOG as far as the logic involved. So you have different types of risk dimensions, and then you have different levels of risk within those dimensions. And like Thor was saying with the filter, so there's all of these things that come into play where we know that it should make it a higher or lower risk. Like with the filter, the surface area there is really, really huge. So you have like a very high surface area to product ratio, things like that. But let's just like step through this and talk about the ones in 1665. So first of all, you have the duration of contact. This really means the duration of contact of the substance or the product with the part, not necessarily the duration of contact to the patient, right? And so if you have something, let's say like a bottle, then that would be something that would have contact for a pretty long time because it could be stored for greater than 7 days. So if we had a bottle, we would say, okay, for duration of contact, that's a level 3. Let's say it's put into the refrigerator, then that would make it a level 2 for the temperature of contact. If we're talking about some organic reagent that's going to be used in the process, then this would be a highly organic thing. And then let's say we know what the bottle is made of. It's probably like a high density polyethylene type bottle. Then if we're really familiar and know all about the it's a type of material for a bottle that's been well used for a long time, then we would say that would be low risk. So you go across these risk dimensions, and based off of the level of detail there, you assign it to a risk level for that dimension. And then you chart this out, and you can use that to assign an overall risk level for the piece altogether. So here is the scoring chart from 1665. So you can see there at the very top, if you have 4 things that are level 3 and there's no additional qualifiers, then that would be the highest risk that you could have. For what we had just a second ago, there were 2 level 3s, 1 level 2, and 1 level 1. And so that, you can find that here. It says, okay, 2 that are 3, 1 that are 2, 1 that are 1. So we're there where it's showing the red circle, and it would be a level B, a moderate risk, or a level C, low risk. It's really small on your screen, but there's 2 superscripts there, A and B, and it says, okay, if the level 2 is in temperature, solvent, or duration, then it should be level C. Otherwise, it would be level B, right? And so, so here we can see, yeah, it was in the temperature dimension, and so it should be this level C. So that's how you kind of like navigate through this. One of the things that I really like about USP 1665 is that after you've assigned a level, then it means that you should go back and decide if there's any mitigating factors that help to change the risk level from a higher level to a lower level. So you have the opportunity to reduce the level of risk by doing this. And so what this is you can see here. So risk is reduced if the potential extractables will be removed from the drug by a downstream process. So if we have a higher risk situation, let's say that it's a risk level B, but it's pretty far upstream, and there's like a downstream filtration process that we know should remove those extractables, then that obviously mitigates the risk, and that would bring it down to the lowest risk level where we would just do something like NVR and a UV. The other thing that it considers is, and as a toxicologist, this matters a lot to me, is that it considers the dose to the patient, right? So in the end, the dose makes the poison. It really matters a lot if the drug is going to be something that's used every day for the rest of the patient's life, or if this is like a one and done type thing, like a vaccine where it's just going to be a single exposure. So If it's a solid or liquid oral type dose, then you can lower the risk of the part by a level. And that makes sense, right? Because we know oral absorption's a mitigating factor. When you take something orally, then it first has to pass through the liver, and that cleans it up. If it's topical, that can be lowered a level. And then the duration of exposure, like I said. So if it's less than 7 days cumulatively, then you can lower the level. So there's all of these common sense sort of ways that we know that the risk could be lowered, and USP 1665 allows you to account for that. So let's just go through this using the same type of device that Thor talked about. So we're talking about a sterilization filter. This is right at the end of the line of a drug process. It's right before it goes into the filling system. So we're gonna, if we consider that the drug is a small volume parenteral, single dose, organic composition, then we can kind of like go through this process. So the duration of contact is less than 24 hours. So the filtration process for the drug is going to be very short. We know that. And we'll say that this happens at ambient temperature. That makes sense. We said in the description there that it's an organic composition. So we're going to call that organic. And then the chemical composition of the components. So most sterilization filters, actually, we do really know the composition very well. But let's say that we were unable to get great information about the composition of this filter. So we would call that a high risk. So we have a 1, a 2, and 2 3s in this case. If we take a look at our chart, that again puts us where we have 1, 2, 2 3s, and a 1. So it's a level B, a moderate risk, or a level C, lower risk. And then we know that the level 2 was a temperature, so that means that it should be a level C. And then we start to ask ourselves, okay, are there any of these mitigating factors? So we say, yeah, it's not upstream to a removal step because it's right there at the very end. But because the drug is only used a single dose, that means we can lower it a level, and that puts us squarely here in risk level B, right? So risk level B, that's 1 extraction solvent in 50% ethanol, and we're doing extractable. So to me, that sounds like a pretty reasonable approach for this type of device, especially considering the risk of the drug product itself. So we start to compare extraction, let's say, strategies, right? So chemical evaluation strategies, let's say it that way. And we can line this up, BPOG versus USP 1665. So for this case, where the drug product is a small volume parenteral, single dose, organic, and we're talking about the filter, this sterilization filter right before it goes into the container closure system for the product, then we would say, okay, both BPOG and USP 1665 would initially call this a high risk. So we'd say, yeah, it's right there at the end. It's high surface area. It's right before it goes into the vial. It's high. But, USP 1665 gives us the opportunity to consider the dose, how the product is actually used and how it interacts with the patient. And when we consider that, we say, oh, wait a second, even though the filter has lots of contact with the product itself is only used 1 time. It's a single dose. And that gives us a mechanism to reduce the risk level from high to medium if we use USP 1665. And that actually, in the end, makes a pretty big difference in the testing strategy. BPOG, it's known for being very very conservative. That's one of the things that a lot of people like about it, is that it's so conservative. For this type of product, you would end up extracting the filter in at least 4 solvents, depending how you read the standard. You might even add a couple more. This would be, purified water, 50% ethanol, and then a high and low pH solvent. Per USP 1665, we would focus in on just 1 solvent, 50% ethanol. The extraction temperatures would be the same, but for BPOG, the extraction durations would include a 7-day extraction in addition to a 24. So you can see same product, evaluated two different ways. Both are using a risk-based approach. However, the testing recommendations that fall out would be pretty radically different. So for BPOG, you'd end up doing 8 separate extractables tests, 4 solvents at 2 different time points. For USP 1665, it would be 1 solvent at 1 time point. A question that often comes up, USP 1665 is still in draft form. It's not yet fully finalized. So which one should we use? Which one should our customers use? It's my opinion, and I would be curious to hear what Thor thinks as well. So USP 1665, this is a consensus standard. It's highly recognized and respected. I think that if I have to choose between BPOG and USP, it's going to be USP. I think this is going to receive much wider adoption in the future. Still, BPOG it covers what USP would have covered, but it goes a lot further. So I think it depends a little bit on the level of conservativeness of the company that's doing the testing, because it just includes a lot more. Thor, do you agree with that perspective on USP versus BPOG?
I totally agree. I think it does come down to the client and their location. If obviously USP is United States pharmacopoeia, so and it is a very well established standard, so it is widely accepted worldwide. But still, it's United States pharmacopoeia. So if you're outside the United States or you do business outside the States, maybe BPOG is more something to consider, but it is definitely the worst case. But those are the considerations you would want to take into account. Yours and what you said too, Matt. 100% agree.
Yeah. Okay, cool. I think that's good. I want to throw this up here. This is our contact information for Thor and I. We certainly are happy to take your questions or talk about this stuff any time. I don't know. It's kind of a battle who is nerdier, Thor or me, but we really do love getting into the nitty gritty of some of this chemistry testing, and also I think we're both true fans of using a risk-based approach. I want to plug for just a second. We have lots of educational resources throughout the spectrum. So this little tidbit has been about using a risk-based approach with single-use systems, but we have a lot of resources for all sorts of other questions and problems over the Sotera Health family. I want to point out our Nielsen Labs, our expert lab testing and advisory services. So we just launched our new Nielsen Labs Academy that has like a wealth of resources that you can use to learn about all different angles here. And we have lots of information on sterilization solutions as well. So with that, I think that we'll go ahead and close. Please feel free to reach out to us anytime if you have questions about this. We would love to chat.